to start a unit now, last one on this chapter on immunity. We're not going to cover it in as much detail as the book. If you're going nursing or you're going to take microbiology for whatever reason, you'll have to take it as for nursing. You spend a lot more time on immunity than we're going to spend here. We need to get on to other things, so we're only going to spend a short amount of time on this. And it can get really involved. All right, so let me lay out specific resistance, also called immunity. You see a bone. This is supposed to represent bone marrow. The bone marrow, as you can see, red bone marrow, is making the stem cells right there that will become all of the red cells and the white cells and so on. And you see that eventually you get these B cells and you get T cells. And notice that there's a shift from bone marrow of some of these cells over to the thymus for processing. Remember, the thymus makes a hormone called thymosin, and it's for processing of these T cells, or T lymphocytes, T for thymus. The B cells are processed either in bone marrow or liver or a couple other places, but you can remember B for bone marrow, even though both of them actually are produced in bone marrow. Now, that leads us to a key thing here. There are two immune responses that you have working for you. The cell-mediated immunity, or cellular immunity, and the antibody-mediated immunity, or what's called humoral immunity. You have a double immune response. Keep that in mind. We're going to go over each one of them. Okay, first, acquired immunity. Here's a layout of the different kinds of ways that you can acquire immunity in your life. We're going to talk more about this in lecture. Active. You come in contact with the antigen, foreign antigen. Your immune system sees it and reacts, and you mount an immune response. You make antibody. You make activated B cells, T cells, blah, blah, blah. You can either come in contact with the antigen by natural means, you don't know about it, or you're given it purposefully, like in an immunization, which has got the microorganism in it. The antigen is given to you on purpose. That's artificial. In passive, you are given an immune response from some other source. Either it comes from other people, or it may come from other animals. For example, they get a lot of antibodies from horses. That's passive. So you're exposed to, let's say, chickenpox or hepatitis B, and you are given immune globulins or gamma globulins or antibodies from other people. You're not making them yourself, you're giving them passively. That's passive acquired immunity. If you're given them on purpose, which fits the scenario I just gave you, it's called passive artificial. If you get them naturally without you having any say-so in it, it's called passive natural, as in mother to baby or mother to fetus. And there are advantages and disadvantages to both of these, because in active acquired immunity, where your immune system sees the antigen, you maintain that recognition for a lifetime. Whereas if you're given immunity passively from another person, from an animal, that passive coverage disappears in a few weeks. It's not long lasting. But the advantage of passive is that you've got it immediately, whereas active acquired immunity takes a few weeks for your own immune system to kick in. So each one, sometimes you're given both of them, or they occur together. Okay, so let me lay out immunity. I've used the word antigen a lot. You already have seen self-antigens. Those would be like A markers and B markers and Rh markers on your red cells. Those are self-antigens. We're more interested in non-self-antigens. Those would be like bacteria, viruses, protozoa, all sorts of microorganisms, or antigens on an organ or on cells, like patient A needs a kidney, and they get a kidney from patient B, And patient B's kidney is going to have different markers or antigens on the cells than patient A's cells have on their kidney. So, non-self. Alright, so some key things. Your immune system is able to differentiate between what is you, what is not you. That's really important. If it couldn't tell the difference, you wouldn't have much of an immune system. Second, it's extremely specific for a particular antigen. So, for example, antibodies made against E. coli or staph bacteria are not good against other bacteria or any other virus or any other microorganism. It's very specific. Once your immune system sees the antigen, recognizes it, it will last for years, sometimes even a lifetime, like some vaccines are good for a lifetime. Or if you get plague, for example, you have a lifetime resistance to plague. Okay, so I've already laid out the two kinds of immune responses. Antibody-mediated immunity, sometimes also called humoral immunity, and cell-mediated immunity, sometimes also called cellular. Okay, you're covered by double immune responses. Okay, so the cells involved. Let's go antibody-mediated. B cells. Alright, so here's a B cell right there. By the way, B cells can also phagocytose antigen. You can see the antigen, a bacterium, right there. It attaches to the antigen by a recognition molecule, a type of antibody, a particular type of receptor. And this B cell now activated, turned on, and it's going to divide into daughter cells, all of which will identify that particular antigen. Its daughter cells will eventually make antibodies specific for that antigen. Now, T cells. Oh, by the way, just to go along with the antibody, the picture next to it shows antibody, and here's that specific antibody made against whatever the antigen. You can see it attaches to the antigen. And basic antibody doesn't really kill. What it does is it inactivates the antigen and immobilizes it so that macrophages and dendritic cells can come along and phagocytose the antigen more effectively. Alright, now T cells. Well, to go down to this picture down here at the bottom, here's the antibody-mediated immunity that I was talking about earlier. Alright, now look over here at cell-mediated. So, look at the T cells right there. They're the ones involved in cell-mediated immunity. Now, there's an exception we'll talk about in just a minute. By the way, the picture you're looking at right here is an electron micrograph, color enhanced. The bacteria, or the antigen, is in gold. They're actually streptococci bacteria, for those of you that have had micro. M stands for macrophage. That's this big gray spreading cell. That's one cell. And the pink guy is a lymphocyte, showing you how they all kind of come together and interact. Okay, so the key guy here, or guys, are the APC cells, dendritic cells and macrophages. You see that right here. Now remember, you've got a phagocytic cell, macrophage or dendritic cell. You've got an antigen, whatever that is right there. Phagocytosis occurs. This thing is taken in, it's destroyed. Pieces of it are now inside of this phagocytic cell. And one of these pieces, or more, it could be more than one. It's hard to use the animation feature or the annotation feature like this. Pieces of the antigen are actually put outside of the membrane. That's what I'm trying to do here, is dot these proteins on the outside. These are antigen proteins on the outside. And this APC cell now presents the antigen to either a T or a B lymphocyte. That's why he's called an antigen-presenting cell. Okay, we'll talk about the different kinds of B cells coming up here in just a minute. So on the picture, again, here's your APC cell. It's coming in contact with the helper T cell. Very complicated receptor recognition setup here involving the antigen. The helper T cell now can become activated, and it can talk chemically to a B cell, and it can talk chemically to a cytotoxic cell, which then either activates cellular immunity or humoral immunity. So you're going to hear a lot more about helper T cells. Helper T cells are the pivotal middlemen. They're mainly part of cellular immunity, but they can play a role, an important role in humoral immunity because they can interact with B cells. Okay, so here's what T cells can do. All right, so let me take you through that. All right, so here we have in blue our APC cell, and there is your antigen being phagocytosed right there. And there's the destruction of the antigen right there. And notice this little blue thing right there. There's a piece of that antigen stuck on the outside of a APC cell. Now, I don't want to get into MHC. MHC, I'll just point out. The yellow molecule, now you can't see it on this cell right here, but it's right there. It's attached to the antigen piece, and you can see it right there also. These are recognition receptors on the APC cells that help the T and B cells recognize the phagocytic cell with its antigen. It's for recognition. All these receptors are for recognition. In any case, if you look down here, here is a T cell. This happens to be a cytotoxic T cell, and over here is a helper T cell. These two guys come in contact with the antigen presented by APC cell, and they make chemicals. You see the chemical molecules as pink and green. These chemicals are the chemical SOS messages among the cells. The helper cell activates a cytotoxic cell, which now becomes activated. It undergoes mitosis, and it differentiates, turns on to an active cytotoxic T cell. And it goes out, carried around by blood or in lymph nodes or wherever, and it comes in contact with a cell that has the same original kind of antigen on it, that little blue antigen. And it's going to destroy that cell. That's why these guys are called cytotoxic T cells. Cytotoxic means kills cells. So again, you can see that the helper cell was pivotal in activating cytotoxic cells. By the way, cytotoxic cells can make chemicals that activate helper cells too. Okay, so here's the plan. You got the antigen. It's phagocytosed by the APC cell. It's presented to a helper T cell. Helper T cell, cytotoxic T cell come in contact. Cytotoxic T cell is activated, undergoes mitotic division, gets turned on into activated killer cells, cytotoxic killer cells. They can either directly destroy the antigen when they come in contact with it, or they can make chemicals that destroy the antigen. The picture that you see over here, this guy right here is a cytotoxic T cell. This guy right here is an infected cell with viruses inside of it. There is the kill by the cytotoxic cell, and there's the total disintegration of the infected cell. By the way, two more things. There's one other thing I need to make reference to. We're going to talk about again in a minute. Memory cells. Some of the daughter cells of the mitotically active, turned on cytotoxic T cell will become memory cells. They float around in your blood and they're in your lymph nodes, and they carry the memory of the antigen for years and years. I am not going to go there, but you might want to look at this animation. It's an excellent animation on cellular immunity. Okay. So let's talk about antibodies, which are going to come from B cells. We'll talk about the B cells and how they make them in just a minute, but let's start with the antibodies. Okay, this is humoral immunity. There's no antibody made in cellular immunity. Um, Look at this molecule right here. Okay, this is one antibody molecule. It's got 4 chains. These are polypeptide chains made of amino acids. You've got these big chains. They're called heavy chains. They are duplicates. And then you've got these light chains, one over here and one over here. And up here at the top where a light and heavy at the end, this is the antigen binding area, and you've got an inset showing that right there. Now, there are 5 classes of antibody. It depends on the type of heavy chain. Okay, so Ig stands for immunoglobulin. It's the same thing as gamma globulin or antibody. They take the I, they take the G, and then they use a Greek letter for the type of heavy chain: alpha, epsilon, mu, delta, and gamma. So you get immunoglobulin G, IgG, immunoglobulin D, IgD. Now, I'm not going to get into all 5 types, but I want to mention 3 of them. IgG, why important? It's the biggest group of the antibodies. About 80% of all your antibodies, IgG. It is also the antibody that they use a lot in serological tests to determine if you have a viral infection or whatever. Very often for viral infections and parasitic infections. It's also the antibody that crosses the placenta in the last trimester. IgA, alpha, heavy chains. It's a secretory antibody. You have it in mucus, you've got it in urine, you've got it in breast milk, you've got it in sweat, you've got it down your respiratory tract in mucus. I mentioned breast milk, so this is the other protective antibody for the newborn via breast milk. IgE, I'm going to mention it fleetingly when we talk about allergic reactions. It's involved in allergies. So, you already have some experience with antigen-antibody reactions. Blood typing, there you go. So remember, we had a blood, well, let me rephrase that. A type of blood, whatever type, we added a drop here, a drop here, a drop here. Then we added anti-A, a drop of anti-B, a drop of anti-D or RH, and then we look for agglutination. And you can see there is agglutination there, agglutination there, agglutination there, making that blood A, B positive. So one of the things antibody can do is it can agglutinate antigen. In fact, that's what you're seeing right there. That's what's pulling the red cells into these funny little clumps. Here's another example right here. This is a monospot test for infectious mononucleosis, or the Epstein-Barr virus. And you can't see it as well as the blood typing down below because there's no red color. You're using plasma from the patient to determine whether they have antibody, and you use a known antigen, and right here you can see positive agglutination and no agglutination, so it identifies that the person has mono. And there are other types of antigen antibody tests, but those are 2 major ones. Okay, humoral immunity. Look at the picture over on the right. I'll take you through it. And again, you got a step by step, and you'll see it's very similar to the cellular that I just took you through. All right, get rid of that. All right, so we start over here with the antigen, and it gets picked up by a APC cell, a macrophage, a dendritic cell. Remember that those 2 guys don't necessarily have to be involved for B cells, because B cells can identify the antigen and phagocytose it directly. By the way, AG stands for antigen. So B cells can phagocytose on their own without dendritic cells or macrophages if need be. Anyway, they are presented with the antigen, or they phagocytose and come in contact with the antigen that way. Here is the APC cell with the antigen. And notice that there's a T cell right there, and it recognizes antigen, and it makes chemicals right here, which talk to a B cell, and this B cell now becomes activated, and it undergoes mitosis and becomes activated, making a population of memory cells, just like the T memory cells, and some of the daughter cells become plasma cells. They just mature, and they make tons of antibody, and that antibody goes out into the blood. And there are those antibodies right there. And that antibody is specific for the antigen. It comes in contact with it, it clumps it, just like I showed you before, making it easier for macrophages to scoop up, or dendritic cells to scoop up the antigen. Really good website for a nice animation on humoral immunity. One last thing. Remember, APC cells don't have to be involved, and even helper cells don't always have to be involved. So the B cell can kind of do its own thing, come in contact with the antigen, and become activated just that way. But usually there is an intermediary. And again, here's a look at the interaction between B cell and helper cell. And those little black receptors on the outside, that's where the antigen would attach to. And it then activates into a plasma cell, which makes antibodies. Okay, primary versus secondary response. Look at the blue line. All right. So let's say you come in contact with, I don't know, bacterium X. First exposure you've ever had. First exposure. You're not going to have any immunity. You're not going to have any activated T cells or B cells. Your immune system's never seen it before. So notice how long it takes to get to the peak here. That's about 10 days, is it not? Well, by that time, you're already going to be sick. The time between contact and your peak immune response, that time period right there, is 7 to 10 days. In fact, you could already be dead if it were plague, because plague can kill you in a couple of days. All right, now let's assume that you get better. And over time, months and years, your immune response declines, it goes down. And some time down the way, maybe a year or 3 years or 5 years, you come in contact with that exact same antigen again. But look what happens now. Remember those memory cells? Memory cells. Those guys up there, they carry the memory of the antigen. So if they come in contact with that antigen again, they immediately activate. They start making antibody very quickly. You get a huge, intense, fast immune response. This is called the secondary immune response. The first response is called the primary response. You may be thinking, well, unfortunately, you could die before you ever really have a good primary response, because it's pretty puny even when you have it, right? Well, I guess that's the breaks. You just die. But if you don't die, you now are going to have a really great secondary immune response the next time you come in contact with the antigen, which is protective. This is the protective immune response. Unfortunately, you've got to have this non-protective primary response in order to set you up for the secondary. Now, look over here at the red line, and you'll see here's a primary immune response to a totally different antigen. Remember? Specificity. By the way, this could have been, I could have given you an example of a vaccination here. For example, let's say here's your first tetanus vaccine, and you get a primary immune response. You know, you get many boosters of tetanus vaccine in your life, and here's a second tetanus vaccine, and you get this secondary immune response. This is also called the booster response, by the way, which is why many vaccines are are given sometimes to boost your immunity to a particular thing. Okay, just fleetingly, immune disorders. Again, you're going to have this in much more detail in microbiology. Autoimmune disease. Your immune system reacts to autoantigens, self-antigens. They could be antigens on your thyroid gland. They could be antigens on sperm. They could be antigens to the synovial membrane. They could be antigens on the outside of cells. They could be antigens on DNA. The autoimmune diseases vary from not so severe to very severe. Look at male infertility. That's not going to kill you. It's just going to make you unable to have children. Men actually make antibody to sperm. But down towards the bottom, there are two very severe autoimmune diseases, lupus and rheumatoid arthritis. And these are really debilitating, life-threatening, particularly lupus, autoimmune diseases. And there's lots of causes I'm not going to get into. Viruses have been shown to be an activator of autoimmunity. Your immune system not seeing antigens until years, self-antigens until years down the way, like with sperm. Sometimes chemicals or trauma, physical trauma can activate autoimmune disease, like breast implants. So there's a lot of different things that can do it. What do you do about these things? Well, some of them are treatable, like Graves' disease. That's an autoimmune disease involving your thyroid, which is actually pretty easy to treat. They just nuke your thyroid cells with radioactive iodine, and you don't make T3, T4 anymore. You don't have any thyroid cells to make it, and you just get put on T3, T4 for the rest of your life. So that's pretty easily treated. But if you go back down to lupus, there is no treatment, really. There's no cure. You're just getting treated for symptomology, and eventually, unfortunately, it gets you. The little girl that you see here in the picture has the typical lupus rash that a lot of people have. It forms a kind of an odd band rash across your face. Okay, hypersensitivities, also called allergies. These are allergies. The picture you see up at the top is goldenrod. A lot of people are allergic to it. And down at the bottom left, you see different kinds of pollen. Some people are allergic to goldenrod, and some people are allergic to mountain cedar, and different kinds of pollen. All right, so an antigen that induces an allergic reaction is called an allergen. And this involves humoral immunity. So if you look at the picture over here, it's the same scenario we've gone over, with just maybe a couple of differences. So think about what's happening here for a minute. First exposure, I don't know, let's say goldenrod pollen. All right, so there's your pollen right there. You inhale it. It comes in contact with macrophages. Your APC cells are going to phagocytose it, present it to T helpers that activate B cells. B cells become activated to plasma cells. The plasma cells make, in this case, IgE. Now, this is all first exposure. Remember, maybe 7-10 days have gone by. No symptoms. You have no symptoms. Okay, so the IgE molecules now attach to basophils or mast cells. Remember those guys? Inflammation. Those little black granules, what's inside of them? Histamine, serotonin, bradykinins, other chemical mediators. Except nothing's happened yet. The IgE is just attached to the outside of the cell. And they're floating around in your blood. No problem, no symptoms. You don't know anything is going on. Okay. But now, second exposure, or third exposure, or fifth exposure. There's a new pollen antigen. It gets into your body. It comes in contact with this guy right there. Remember, that guy already has IgE on the outside of the molecule. Right there. See those little Y-shaped molecules right there? They're already there. They attach to the antigen. The whole thing explodes, dumping huge amounts of chemicals. Really severe inflammation. Okay? Redness, swelling, pain and discomfort, heat, but in addition you get airway constriction, mucus secretion, closes up your airway if it's a respiratory allergy, or you can have hives on your skin if it's a ingested allergy. Things like pollen, food allergens, particularly peanuts, um, cockroach, fecal antigens. Some people are allergic to dust mite feces, mold spores, all sorts of things that can be allergens. And last, I wanted to point out HIV, although you're going to have it a lot more in microbiology. So you see the virus at the top left. You see the virus in a picture at the bottom right. The difference, of course, is you're looking externally at the virus here and you're looking internally over here. See those knobbies? Okay? These are the things that you see right here, right? They are receptor structures that the virus uses to attach to T helper cells. If the cell does not have the appropriate receptor for these little viral attachments, it can't get into the cell. T helper cells have the right receptors, and some macrophages and dendritic cells also do. But the main host is a T helper cell. So you can just imagine what's going to happen here, because you know the function of T helper cells, right? Now on the inside of this virus, I'm not going to get into all this because you haven't had microbiology where you go over viral structure. But there's viral genetic information right here in red. You see it right there. And this genetic information gets into a host cell. Okay, so here's a T helper cell right there. And the viral information now either codes for new viruses inside the cell, or the viral genomic information can actually become part of the host cell chromosome. This is supposed to be DNA inside of the nucleus here. And the cell can be infected for years but not make viruses. Eventually, the cell will start making baby viruses. The cell disintegrates, baby viruses released to do the whole thing again. So, you get a depletion of the T helper cells, and obviously, T helper cells are critical in cell-mediated or cellular immunity with cytotoxic cells, but remember they're also the pivotal helper guys for B cells. So you can see what this is going to do to your immune system. Okay, so I've taken you through cell infection. Symptoms, basically this undercuts your whole immune system, making you extremely susceptible to other types of infections, other viral infections like herpes, or yeast infections, fungal infections like candida yeast, or something as simple as E. coli that everybody has, or staph bacteria, or just the most common types of microorganisms that you've all come in contact with can kill people that have AIDS. Another thing that these people get is cancers, because cellular immunity surveys for cancer cells and gets rid of them. So these two pictures have a type of cancer called Kaposi's sarcoma. It's a type of connective tissue cancer. It was usually only ever seen in older men of Middle Eastern ancestry, but in the 1980s it was seen in men in their 20s, 30s, 40s. It was very odd to see that in those people, and that was the first indicator that there was a new disease. Difference between HIV and AIDS, again, not going to get into all of this. There's some criteria that define AIDS. One is that you have less than 200 T helper cells per cubic millimeter of blood. You should have over somewhere around 1,000, maybe 800 to 1,200. So this is a real drop in your T cell count. And also in AIDS, you get a lot of opportunistic microbial infections that I mentioned earlier, including tuberculosis, which is a real problem in these people. And that is it.